here. Anyway, I will try my level best to understand microscopy. Yeah. So first of all, you should know few important things. Types of microscopes. Types of microscopes. I think I will draw the classification. Everyone go through this one. Types of microscopes. So first I will discuss microscopes are divided based on their source of light, placement of light, then type of light used, placement of light, type of light, source of light, and also based on their property of light, and property of light and measuring of light. So see, everything is light about microscopy. As C. Raman started his career on light, so we will go with that same light spirit. So as I stated, based on sources of light, microscopes are divided based on the placement of light, types of light. The types of light means polarized, plain polarized light, then unpolarized light, refracted light, reflected light, all the properties. Okay. So yeah, again properties of light. Is it a refracted? These properties and measuring of light. What light we are measuring? What type of light we are measuring? So based on this, the microscopy are classified. So first I will start with two types of microscopes: light microscope and other one light microscope and other microscopes. I can simply say other microscopes. That means other than light, what thing we are using to measure the specimens. If we are not using light, we might use electrons. Hence we got this electron microscopies. We have scanning electron microscopies and transmission electron microscopies. So I am not discussing about electrons, rather than I will focus on light microscope. Okay, where we are using light as a source. Okay, so coming to the light microscopy, there are two types: bright field microscopes, and dark field microscopes. What is this bright field and dark field? Can anyone say what is the meaning of bright field and dark field microscopes? No idea? Listen students, the major difference between bright field and dark field is the meaning field. Field means background. What background we are using? Is our background bright background or is it a dark background? Based on that, we are classified light microscope into bright and dark. I will give one example of bright and dark. So first I need a schematic diagram of basic microscope. So tell me the parts of microscope. We have specimen collecting area, right? Yeah, this is stage of the microscope. Then below the stage there is a lens. This lens will there is a mirror. This mirror will capture the light, and light will go to the first condenser. So condenser and diaphragm. There is a diaphragm. What is the significance of diaphragm? It will adjust the amount of light. The pupil of the diaphragm will dilate or it will constrict. So diaphragm will adjust the amount of light. Then there is something called condenser. What is the purpose of condenser? Condenser will condense the light. Okay, this bright light will be condensed onto it will focus onto the specimen. Okay, it will focus the light onto the specimen. That is the purpose of condenser. Okay, now condenser. Then here we got a specimen. Light transmitted on the specimen. Then whatever came through the specimen will be collected into the objective lens. It will be collected into the objective lens. This inside the objective lens there are three types of lenses: 10x lens, 40x lens, and 100x lens. Okay. So this lens will collect the light again, and it will focus on to the eyepiece lens. And eyepiece lens, here we will see the thing. This is eyepiece. Then this is objective eyepiece lens, objective lens. Then condenser. This one. These are the basic parts of the microscope, and they are connected like this. You know, right? That's how they are connected. Anyway, now the lens position you understood. Now see students, in bright field, the light has been passed on to the specimen. Imagine the specimen is a transparent specimen, amoeba. I didn't stain the amoeba, and I kept the amoeba solution with amoeba on the slide, and I sent a light through it. Can we able to see amoeba? We cannot see amoeba. Why? It is transparent, and the background also white, so we can't see it. So in bright field we cannot measure. In bright field only stained specimens can be observed. Only stained specimens will be observed in bright field. Stained, it will be pink color, blue color, gram staining, right? Only stained specimens can be seen in bright field microscope. The background is white or bright. So bright field microscope is good for measuring or studying the stained specimens. But stained specimens will come with a cost. We are killing the specimens. We are killing the microbes. So we cannot study the motility. We cannot study motility in a bright field microscope. Am I correct? So is there any alternative to study the non-stained specimens? Non-stained motile living specimens. Is it possible? Yeah, it is possible. What we did is, if I keep a dark background, imagine you have some glass sheets, glass doors, okay? And this room is dark. We switch off the lights in the room. So some of the light from outside will hit the glass, and I can see the glass in a dark room. But I cannot see a glass in a bright room, because light passing everywhere and it looks very transparent. Similarly, I will keep a dark film in the condenser, and I will pass the light. When I pass it light through a dark film, now the background became dark. Okay? Now the specimen will looks like a ghost. Some of the light will escape from the specimen boundaries. So we will see a bright specimen moving here and there. That is the idea of dark field microscope. In dark field microscope, we are introducing a small film, dark film, in the condenser, so that we can able to see a motile and living organisms. That is the purpose of dark field microscope. Is it clear? Yeah. So yes. So that is where I said the placement of light. We can say not placement of light, that will come later. So up to here is clear, right? Then source of light. Rather than using a normal light, we can use UV light also. For what purpose we will use UV light? Any idea? For what purpose we will use a UV light as a source in a microscopy? Any idea? Listen, the idea of UV microscope is, I have a tissue, I stain the tissue with a fluorophore. If I hit a UV light on the fluorophore, it will give a green, bright green light. Right? So that means fluorescent microscopy. In fluorescent microscopy, I will use a UV light rather than a normal light. In fluorescent microscopy, I am using a UV light. Okay? Now listen. Tell me the position of the UV light in fluorescent microscope. Where I will place the UV light? Shall I keep UV light below the specimen, or I will keep UV light above the specimen? What is the ideal position of UV light in a UV microscopy, fluorescent microscopy? Shall I need to keep UV light at the bottom of the specimen or top of the specimen? Yes? Any guesses? It should be kept on the top of the specimen. Then only UV light will hit the specimen and it will give the transmitted light. That's why in UV microscope, the UV light will be on the top of the microscope, not at the bottom. So now we understood placement of light and type of light. Okay? In UV microscope, the light is on the top of the specimen, not in the bottom. Now we understood. Am I right? And what is the purpose of UV light? To study the fluorescent microscopy, fluorophore components. That is the idea of UV light. Then, there are other interesting things about other microscopies. I will tell other one. Now I will discuss about the polarizing microscope. Polarizing microscopy. Polarizing microscope, there is one more microscope. Through your notes, tell me students other types. Polarizing microscopes, then? Contrast microscope, then? Light microscope we discussed. Inverted microscope. Inverted microscope, okay. Inverted microscope. Okay. One second. See, that's why preparing to the class is very important. I know. I will give that. Yes. 1 second, class. Phase contrast microscope improves the contrast between this a special optical lens. A cover. We'll see students, 1 second. I will finish first the normal light microscope types. Now, the light microscope are many types. This microscope has further types. How they classify this microscope is based on head, based on the eyepiece. Based on eyepiece, this light microscope are divided into 3 types. If it has only one eyepiece, that is called as mono, that is one ocular eye, medical term for eye is ocular microscope. Monoocular microscope. If it has 2 eyepieces, then it is called as binoocular microscope. Then, is it possible to have multi heads to a single microscope? Is it possible to measure a single specimen from a single microscope by 10 students? It is possible. That is called as multi-headed microscope. Multi-headed microscope. This multi-headed microscope, you can Google it, you will get it. Okay. It's a stand with multiple eye lenses. Okay. It's a modified microscope. Yeah, fine. Then refractory effects. Coming to the phase contrast microscope. Phase contrast microscope. So the idea behind phase contrast microscope is like this. Do you remember what is the refractive index of a glass light? Can anyone say? Light will bend when it is moving from air to a solid medium, right? That bending of light is called as refraction. What is, we have standardized the refraction of a glass light, the microscope glass light. That is 1.4. The refractive index of a glass light is 1.4. The bending of light will be 1.4. Okay? Now listen. This refraction plays a vital role. Refraction of light plays a vital role. I will tell you how. Your microscope lenses, objective lens. Your microscope objective lens was fixed at 1.4 Ra because it can only detect 1.4 Ra my slide light. That means light came here, it's slightly bended, and the bended light only it can detect. It has been standardized, these two values. Now let us see one problem. I have a transparent specimen. Transparent specimen has many elevations. Each elevation has their own refractive index. Maybe 2, 3, 4, 6. Each elevation has their own, bended light. Can I detect that bended light in normal microscope? I cannot detect such light in a normal microscope. Results in blurry image. Why I am getting blurry image? Because the light on the specimen was moving here and there. It's scattered. It's refracted. Now listen. I know that this is a cell. Nucleus, ribosome, mitochondria. Nucleus has a refractive index of maybe 2. Ribosome has 3. Mitochondria has 2.5. And overall cell has 1.4. In normal microscope, I can only see the cell, but I cannot see the cell internal structures. The reason is their RNA is different, and I cannot see the internal components because their refractive index is different. Is it clear? Now, I can do one interesting thing. Below the specimen, near the condenser, I will keep a specialized lens. This lens will bend the light. And there is one more lens near the objective lens. I can take 2 specialized lenses, maybe some specialized mechanism in the diaphragm and in the objective lens. Here what I am trying to do is, I will adjust the objective piece RI to the designated RI. Now I will change the RI of my objective lens. Maybe I will keep RI to 2. Now I will see the nucleus of the cell. Now the nucleus will be prominent in my microscope. But again, if I am changing the objective lens RI, now I get 2.5. Then suddenly mitochondria will appear in the specimen. So by changing the phases of the refraction of the light, I can see the multiple intracellular components in a single cell. This is possible by phase contrast microscope. In phase contrast microscope, I was changing the refractive index of the light. I was changing the phases of the refracted light. Hence, I can see the internal components of the cells. That is the purpose of phase contrast microscope. Now listen, in phase contrast microscope I got it, but still this is a bright field microscope only. I am not staining it. I am seeing a living cell. Internal components of a living cell has been seen with a phase contrast microscope. Again the problem is they are transparent. Can I able to induce colors to my cell without staining the cell? Is it possible? You got my point, right? Can I induce colors to my cell without inducing staining? I want colors to a living specimen without staining. Is it possible? Is it possible to get a colored image from a non-stained specimen? You just need to be Newton's now. What Newton did in 1600s, he took a prism, he kept a light, light was split into 7 colors with Xeon. Right? We had Z-I-O-R. Okay, indigo, blue, green, orange, red. Right? Now listen to me. Can I keep a small prism beneath the condenser so it will split the light into various colors, and then the colored light will hit the specimen to the right RI color? That right organelle will be in a specific color, in a colorful specimen. So now I am using the property of the light. I am using the property of light where it will give 7 colors, and I was transmitting a colored light to the specimen. Now my specimen will be in color colorful way. See how much depth we are doing with the understanding light. Without staining the specimen, now we are getting a colorful images of a live specimen. And that is the idea behind the polarizing microscope. The light has polarization, polarized and non-polarized light. So by using the polarized, there are two types of different mechanisms, polarizer and analyzer. In polarizing microscope, we have a polarizer in the diaphragm and analyzer in the objective lens. Okay. So by changing these two things, we will get a colorful images out of this one, polarizing microscope. You can Google yourself polarized microscope specimens. You will see various things. Or I will say like this. You will see a colorful diamond under this specimen. Okay, if you keep a diamond here, you will see colorful diamond because a diamond has various angles and various angles will give various colors. So crystals can be studied in a polarizing microscope, and crystallized fossils can be studied. Say for example, there is a mosquito in a silicon in the sand. So that mosquito specimen can be studied with this. Or I will say one more thing. I took a fly. It has a wing, and I want to know the layers of the wings. Each layer has its own refractive index. So the layers of the wing can be studied by polarizing and refractive microscopes. That means phase contrast microscopes. So this is their applications. Again, by the way, I just want to tell you one thing. I have worked with the phase contrast microscope in ICMR. The cost is around 70 to 80 lakhs. These microscopes are very, very costly. They are very costly microscopes. So this is not as simple as this. I said we got one device here and here. Actually, there are almost 20 to 30 lenses. So 20 lenses are splitting the light inside a microscope. It's not just a single lens. 20 to 30 lenses splitting. So it's a huge science behind it. So all is possible by understanding light. Okay, again, CB effects. Then light microscopy has their own limitation. The limitation is our eye power. Whatever we will see, that we can see under microscopy. To an extent, some microscopic things can be seen, but we cannot see the molecules. Molecules are very small. So we cannot see molecules with a normal microscope or with light. I will tell one interesting thing. Light travels in waves. Some molecules are smaller than these waves. So these molecules obviously we cannot detect because they are beyond the radar. Got my point? Those molecules are smaller than a light wavelength. So we can't detect them. Then we are using electrons to detect such small molecules. Now I will tell you this one. If I use an electron, that electron, pay attention, I took a specimen. First, I kept the penetration of electrons to 1.5. So they penetrate up to here. And they scan at 1.5 meter depth of the specimen. Then I set electrons at 1.4. They scanned again at second layer. Then 1.3, third layer, 1.2, other layer. So I was scanning layer by layer of the specimen. Then at the end of the day, I will get some ups and downs. This is the idea behind scanning electron microscope. Scanning electron microscope will give the surface structures of the molecules. A viral shape, the surface shape of a virus can be understood by scanning electron microscope. But I want to study the viral internal structure. What is the nucleic acid of the virus? But my electrons cannot penetrate into them. If I want to study the internal components of a molecule, then I need to transmit my electrons through the specimen. Hence, we got a transmission electron microscope. Transmission electron microscope will study the internal parts of a microscopic object, whereas scanning electron microscope will study the surface of the microscopic object. That is the idea behind the scanning electron microscope and transmission electron microscope. In transmission, we are transmitting electrons through the specimen, whereas in scanning, we are just scanning them. just like your scanners, lab scanners. Yeah, this is all about microscopy. Any doubts? I will finish this lecture by one topic, students. Listen. If whenever you are studying any topic, try to explore the topic. Why we are studying it? Is there any benefit after studying it? What benefits I will get as a lab technician? If you can't get answers for this and blindly studying is useless, try to see the applications of these things. Why you are studying polarizing microscope? Why you are studying dark field microscope? Why is there any useful to me as a lab technologist? With that you need to study, students. Alright.